So I'm going to attempt to go through the nitrogen and phosphorus cycle, but relating it to like mycorrhiza fungus and root nodules. So let's do mycorrhiza first. Both these diagrams are showing roots. So, here's a root, and these are the epidermal cells of the root, just like upper and lower epidermal cells of the leaf. Epidermal cells. You can see that some of them are modified into root hair cells. You can see how thick these root hair cells are. They're renowned for having a very large surface area to volume ratio. However, the mycorrhiza fungus is even smaller. Yeah, so this here, I'm going to do it in blue so it doesn't clash with the black line. This is our hyphae, yeah, or mycorrhiza fungus or fungal, and they're called hyphae. Oops, I'm spelling that badly. P. Hyphae, like threads. So this is like a fungal, a fungus made of these really fine threads, and therefore, they increase the surface area for absorption. So in the soil, anything that the plant wants, if it has mycorrhiza, it's going to be better at absorbing it. So let's put something into the soil and zip through this nitrogen cycle thing first. So let's assume that there's some dead plant, and therefore there's going to be some detritus, and that detritus contains protein. So we're going to start with some protein in detritus. Yeah, dead and waste material. Well, that's going to be broken down into ammonia or ammonium ions. And that's due to the waste product, because the thing that feeds on this is the saprobiotic or the saprobionts. Yeah, these are bacteria and fungi that feed on the protein in detritus. And they're going to secrete proteases, and I could, endo, exo, and dipeptidases, and they're going to absorb amino acids, and then they're going to do something called deamination. That's basically they metabolize excess amino acids and they release this waste product called ammonium ions back into the soil as their waste. So this process is called ammonification. Our liver carries out ammonification as well. It's just that we convert it into urea instead of doing that. These ammonium ions don't last in the soil. Ammonia will evaporate really easily, and they're super soluble, so they'll just be lost from the soil. So there's never any really ammonium ions around in the soil, and also these are used by nitrifying bacteria. Oops, I'm in blue water for the name of the process. So we're going to do some nitrification. So this is nitrifying bacteria, and this is where ammonium ions get converted into nitrite ions. Nitrite ions that then go into nitrate ions. So both of these are nitrification steps. So this is also nitrification. And that's carried out by nitrifying bacteria. So this is the end of the road, like in the soil. The nitrates can then be absorbed by the roots. So it's going to be taken up by the mycorrhiza. It's going to be taken up by the root hairs. But you can see by having these mycorrhiza fungus, more of it is going to be transported into the root, and then it can be taken up in the xylem to the leaves, where it can be reformed into amino acids and proteins with the help of glucose from photosynthesis. So all mycorrhiza fungus do is increase the connection of the plant with the soil. So it's only beneficial if you're living in soil that contains detritus. because there's going to be a release of nitrates, and you want to compete with the neighbouring plants to absorb these nitrates. So these tend to be found, if you think of succession, in a latecia. So like in a forest, in a climax community, the plants are highly likely to have mycorrhiza fungus because there's massive competition for the nitrates in the soil. Not only can they take in nitrates, they can also take in phosphates as well. So let's do the phosphorus cycle. So instead of starting with proteins in detritus, I'm going to start with something that contains phosphorus, and therefore, if I had to name things that contain phosphorus, well, there's DNA does, phospholipids do as well. Yeah, both of these contain in detritus, and these are fed on, and then we're going to release the phosphate ions. And this is carried out by, it's the same ones again, it's the saprobionts. So the saprobionts are feeding on these, digesting it. We don't have to worry about what enzymes they use, but yeah, we're releasing these phosphate ions. And then the phosphate ions can be taken up by the mycorrhiza or the root hairs. They're going to get into the plant and they're going to go up here. So in this xylem, I've got nitrates, NO3 minus, and I've got phosphates, PO4 3 minus. You can see that the nitrates, the nitrogen is changing form. It's going from ammonium ions at NH4 plus, NO2 minus for nitrite, NO3 minus, whereas phosphates always stay the same. Phosphorus is really reactive. It never lets go of its oxygens. And therefore, a phosphorus cycle is basically this. And they go up here, and then they're just going to come back onto that side, or they're going to come back onto that side when the plants die. Or if a plant gets eaten, it's the same thing. It goes into an animal. But yeah, it always, in the phosphorus cycle, it always stays as phosphate ions. So there's nothing really to the phosphorus cycle. Both of these, phosphorus, so phosphates and nitrates, can undergo something called leaching, which means they can be washed out of the soil, and they both cause eutrophication. As in, they cause algae to grow faster because the algae can make more DNA, phospholipids, proteins, amino acids, etc. for growth. So yeah, so both of these link to the same kind of like bits of biology. Yeah, the mycorrhiza increase the uptake of both of them, and and they're both related to the problem of eutrophication. And lastly, what does the mycorrhiza get out of it? Well, if you imagine when this soil like dries out, there's like a month of no rain, all these threads are going to die. Yeah, they're in the soil, and the soil's going to dry, and these threads are going to die. However, the fungus inside the roots is protected by the roots, and therefore it can just grow out again when the soil gets wet. So if it was free living in the soil, it would all just die. But it's got like a refuge area within the roots of a plant where it can survive. So the benefit for the plant is that it's got faster access to nitrates, NO3 minus, and PO4 3 minus, for making DNA, proteins, etc. And the positive for the mycorrhiza fungi is it's protected, from drying out. I would write desiccation, but I'm not that confident. It also gains gains sucrose from the phloem. So down here is going to come the phloem, and it's going to supply this with sucrose in the mass flow hypothesis. So we've kind of covered the nitrogen cycle there and the phosphorus cycle. What we haven't done yet is nitrogen fixation. So let's do a root nodule. So this is a similar picture. This is a root again. So this is a root, but it's of a legume stroke. You know, like, in terms of our syllabus, it's a pioneer species. Yeah, a plant that's going to inhabit the soil where there's very few, if any, bits of detritus. Okay, so inside this, and this is our nodule. So it's a growth. It's a growth because in here are living bacteria. So these are bacteria living within this root nodule. So these are nitrogen fixing bacteria. And they're nice and happy in here because put some xylem on. The phloem. So this is phloem. So the phloem connects to it. So this is the phloem. And therefore that's going to supply sucrose to these bacteria. And therefore the bacteria are really happy, and therefore they start to just do this process where they take nitrogen gas, plus they need some water, plus they need lots of ATP, hence why they need the sugar for respiration, and they make ammonia, NH4 plus ammonium ions, so that's ammonium ions. So therefore in the xylem, here's our xylem at the side, this is also like connecting to it, but the xylem's going this way. So phloem's coming down because it's coming from a source to a sink. So yeah, my phloem's coming down, and the xylem goes up, and therefore the xylem's going to take the ammonium ions up to the leaves. It's a fixed form of nitrogen, and therefore it can be used, used to make amino acids, and therefore you can form a protein. So, this is nitrogen fixation. Nowhere are nitrates on this diagram. Nitrates are irrelevant. It doesn't need to form them into nitrates. There's no nitrifying bacteria within the plant. This is within the soil, but within an encased nodule, so it just stays. It's already in a usable form. Within this root as well, just for a bit of extra knowledge, there's like another compartment here, and this is really red. And it's really red because it contains a form of hemoglobin. And that's really basically to kind of like take all the oxygen away from there, so that the nitrogen fixing bacteria, this process only really works in anaerobic conditions. So that's our nitrogen fixation. Again, these are found in early sears of succession, so like a sand dune, so that they can make their own. Again, there'd be no point a plant like this living in a sand dune having mycorrhiza, because the plant's not going to benefit because there's nothing in the soil worth absorbing. Yeah, so that's why these are found on, in like rubbish soil. And farmers sometimes grow these, and then they don't harvest them. They just plough them back into the soil. That increases the amount of protein in the soil, and then I'm into my nitrogen cycle over here. The last bit that we haven't covered in the nitrogen cycle is denitrification. So let's just do that quickly. So then we've got denitrification. So this is where we have nitrates. NO3 minus gets converted into N2 gas. I know it doesn't balance. So where's the oxygen going? Well, that's stolen by the denitrifying bacteria. So these thrive or are common in anaerobic soils because they can still do a form of aerobic respiration using nitrate ions as their terminal electron acceptor. So they are really successful. So yeah, so farmers do two things. They plough to aerate soil. Aerate soil to discourage these. Keeping the soil aerobic will stop these bacteria from wasting the nitrate ions, because I want these to go into the plant. I don't want them heading this way into the atmosphere. You can also increase drainage as well. So if you increase the drainage, it'll keep it aerobic. If you increase the drainage, though, you'll lead to more leaching that can lead to more eutrophication. So it's stuff that really you would have to evaluate. I've got over my 15 minutes, so hopefully there was something there. Again, it's a really useful thing because it ties in with succession, it ties in with the ecological aspects of eutrophication, and people get these wrong all the time. Yeah, so it's just thinking like the bigger picture about them. And yeah, the phosphorus cycle, there is nothing to it. It just stays as phosphate ions all the time.